Welcome to Bio Bits and Bytes. For centuries, the brain has been, the ultimate black box, this impossibly dense, complex thing where our thoughts, our memories, our whole sense of self, it all lives in there.
Right, and we've had maps of it before, but they're more like those beautiful old hand-drawn maps of a city.
You show you the big districts, maybe the river.
Yeah, not the major avenues. It gives you the general layout, but it tells you absolutely nothing about the actual life inside, the dynamic flow of traffic.
That's a perfect analogy.
Yeah.
Because that's the core challenge we're facing now in neuroscience, isn't it?
It is. We know the big neighborhoods, sure, but what researchers on the cutting edge need now are the GPS coordinates.
Not just for the streets.
No, for every single house, every car, and even the utility lines connecting everything underground. If we really truly want to understand how the city of the brain manages its traffic, how information actually flows, we have to move beyond just observing structure.
We have to start mapping them.
Mapping them with data science, with molecular sequencing, and at a scale that was frankly unimaginable even a decade ago.
So that's our mission in this deep dive. We're going to explore that collision of biology and massive, massive data. We're looking at the foundational techniques that combine high-resolution microscopy, really ingenious DNA barcoding, and some serious heavy-duty computation.
All to decode two fundamental things.
Right. First, molecular location, what's known as spatial transcriptomics. And second, the physical wiring diagram, the neural connections, which is called connectomics.
And we really want to give you a shortcut, to getting up to speed on the methods that are going to define neuroscience for the next 100 years.
But first, we have to just sit with the sheer scale of this challenge. It's hard to get your head around.
It really is. When we talk about mapping the human brain, we're dealing with approximately 10 to the 11 neurons.
100 billion neurons.
100 billion. And then a staggering 10 to the 14, maybe 10 to the 15.
Okay.
Synapses or connections. These are just astronomical numbers.
They don't really mean anything until you can ground them.
Exactly. So to make it real, let's look at a recent landmark effort. They imaged a tiny, tiny fragment of human temporal cortex.
How tiny?
1 cubic millimeter. That's about half the size of a single grain of rice.
Half a grain of rice.
And that tiny sample generated 1,400 terabytes of raw image data.
1,400 terabytes.
To put that into context for you, that's the storage equivalent of about 700 high-definition Blu-ray movies.
All for a speck of tissue.
All just to describe that minute piece of tissue, which contained 57,000 cells and 150 million synapses.
That's not a city map anymore. That's a digital universe where you have to catalog every single particle.
That's the mountain we have to climb.
Okay, let's unpack this. 1,400 terabytes. That is the petabyte problem in miniature.
It is.
So it's clear we can't solve this just by building faster microscopes. What are the newest tools researchers are using that, fundamentally change how we collect and process this data? How do we turn all that raw information into actual biological insight?
Well, to solve that scale issue, you need innovation in two different fields at the same time.
Yeah.
First, you need new molecular tools that tell us what molecules are there and, crucially, where they are in space. That's the molecular GPS.
In the second.
And then you need new imaging and computational tools to map who is talking to whom. That's the connectome.
So let's start with that first part, the molecular location, the spatial data. It's really fascinating to me that DNA, the basis of life, is now being repurposed as a kind of high-precision tracking tag.
It's a brilliant pivot. The initial breakthrough was single-cell sequencing. DNA barcodes were used as molecular ID tags to index all the different cDNA libraries.
And that let researchers analyze thousands of cells at once, knowing exactly which cell each transcript came from.
Exactly, but you're right, that was only half the story.
Right, because you knew which cell, but you didn't know where that cell was in the tissue.
Precisely. The next frontier, the one we're diving into, is spatial transcriptomics, finding out where those molecules are in 3D space.
Not just knowing the general cell they came from.
Yeah, and the real challenge there is getting true single-molecule resolution when you're decoding these DNA barcodes spatially.
Because they're so small and packed together.
They're so small and so densely packed that standard sequencing methods where you pool all the molecules together just completely destroy that critical positional information.
So to tackle this, researchers basically had to invent their own machine, right? A combination of a molecular microscope, and a sequencing machine.
They did. They developed this specialized technology, an in-house system they called SMSBS, which stands for Single Molecule Sequencing by Synthesis. They weren't just ordering an off-the-shelf sequencer.
Why not? What was wrong with the commercial ones?
Well, they recognized that the existing commercial platforms, which are amazing for massive throughput, just weren't designed for the kind of spatial resolution they needed.
So they built their own.
They took inspiration from the principles of single-molecule sequencing, systems like Heloscope, but they customized it heavily. They essentially built a bespoke sequencing platform.
What did that look like?
It was a combination of a standard, high-sensitivity microscope, some fluidic control pumps, and this unique 1 microliter volume flow cell that they could easily modify for different experiments. The whole thing was just optimized for one goal: maximizing spatial information.
Okay, so let's talk about that sequencing by synthesis method, because the mechanism is what gives you the location data. It uses something called virtual terminator nucleotides. How does that stop-motion process even work?
The key is that these virtual terminators, VTC, VTT, VTA, and VTG, are specialized. Each one is tagged with a different colored fluorescent dye.
Okay.
So the process works in these very carefully controlled cycles. First, a VT nucleotide is incorporated onto the growing DNA strand.
But because it's a terminator, it stops everything.
It stops synthesis cold.
Yeah.
Then all the unincorporated VTs are washed away, and a high-resolution fluorescence image is taken.
And that single frame, that picture, tells you everything.
It does. The image registers which color dye incorporated at which exact spatial coordinate on the flow cell. So you get the nucleotide's identity and the molecule's precise location.
And then they just repeat that.
Then they use a chemical agent, TCEP, to cleanly snip off both the fluorescent dye and the inhibitory group that stopped the reaction.
Which lets the cycle start all over again for the next nucleotide.
Exactly. That start motion process literally turns molecular synthesis into a high-precision GPS readout. Being able to track a single base pair in 3D space like that is just, it's stunning.
That's the real breakthrough then, turning a chemical synthesis process into a spatial imaging technique.
Yes.
So when they benchmarked this custom SMSBS system, how did it perform? What was the data quality like?
It was highly robust.
Yeah.
They successfully identified 30 different types of DNA barcode molecules, and they got a really acceptable error rate of less than 5% per nucleotide.
And the read length?
Crucially, they achieved an average read length of about 20 nucleotides, which was more than enough to uniquely identify the spatial identity of all those different barcodes.
And pretty quickly, the utility of this expanded beyond just looking at RNA transcripts, didn't it? They realized this system could spatially decode any DNA barcode.
Absolutely. One of the immediate high-impact applications were showing that the system could spatially identify DNA barcode molecules that were bound to antibodies.
At single molecule resolution.
That single molecule resolution.
Yeah.
And that's a huge deal because DNA barcoded antibodies are the backbone of these massively popular methods like CISEC and Codex.
But those methods didn't have the spatial part.
Exactly. Before, you knew which antibody was present, but not where. The SMSBS system adds that critical spatial information to the antibody identification. It bridges the gap between molecular identity and molecular location.
That integration sounds like they found a path toward a new kind of spatial proteomics.
They certainly did. They even dubbed it molecular footprinting.
What does that entail?
It's a method where you physically transfer the DNA barcode molecules that are already labeled on the surface of or inside cells directly onto their specialized sequencing flow cell.
And once they're stuck on there?
Once they're immobilized, you can sequence and spatially analyze them using the SMSBS system. It's a proof of concept that the technology can scale, not just for mapping RNA, but for high-resolution spatial mapping of the entire protein landscape of a cell.
That's a great question. Let's break it down. When you're dealing with single molecules on a flow cell, especially with all the fluidics and wash cycles that are needed for, 2 days of sequencing? Stability has to be everything.
It's paramount. If the molecule drifts even a few nanometers, your spatial data is completely ruined.
So what was the secret to getting that long-term stability under continuous fluidic pressure?
That was a major engineering hurdle they had to overcome. They needed a really effective sample capture method, and they focused on optimizing biotin-avidin interactions.
Which is usually a non-covalent bond, right? Not as permanent.
Right, it's not as permanently linked as a covalent bond. But they found a way to engineer extreme robustness into it.
So how do they manage to make a non-covalent bond hold up for 2 whole days of washing?
They realized that while one biotin-avidin bond might be pretty transient, Multiple bonds happening at the same time would effectively lock the molecule down.
Ah, so strengthen numbers.
Exactly. They engineered their capture oligos to have an increased number of biotin molecules per anchor point. So instead of just one, they used, for example, biotin by 4.
And that did the trick.
That multiplication of non-covalent interactions created a stability that was comparable to a permanent covalent binding method. It was essential for keeping the molecules in place through those long sequencing runs. It's a really brilliant example of using molecular engineering to solve a macroscopic stability problem.
So the SMSBS system is tackling the molecular side by combining sequencing and imaging. But there are a lot of other spatial methods out there that rely only on imaging, and they're facing a completely different bottleneck.
You're talking about the fluorescence in situ hybridization techniques.
Yeah, things like MERFISH, SICFISH, and ISS. Once they get the raw image data from these approaches, what's the major computational headache?
Oh, the headache is a lack of standardization. It's a huge problem. These techniques generate these vast amounts of raw image data, thousands of images per experiment.
And everyone processes it differently.
Traditionally, yes. Researchers have relied on poorly documented in-house code to process, decode, and analyze it. This means if you get data from Lab A running CFISH, it's incredibly difficult to reproduce the results, or even compare it effectively with data from Lab B running MERFISH.
So the core protocols might be open source, but the essential processing logic is just hidden away in these proprietary or poorly maintained scripts.
Which completely destroys the reproducibility of the science.
Right.
So to solve this critical bottleneck, researchers introduced something called pipefish.
Pipefish.
Yeah, it's a semi-automated, generalizable, and crucially open-source pipeline. It's built on an existing library called Starfish, and it performs transcript annotation for all these different fish-based methods.
It's creating a common language.
A necessary common language for spatial analysis. They verified its quality using real data from MERFISH, SEQFISH, and ISS experiments to show that it works across the board.
Let's dive into one specific innovation inside that pipeline. The check-all decoder that was developed for SEQFISH data. What inherent problem in the imaging process was this tool designed to fix?
The problem is something called spot drift.
Spot drift, yeah.
Yeah. CFISH experiments involve many sequential rounds of hybridization, imaging, and wash cycles. Between each cycle, the exact location of a fluorescent spot, which represents a single transcript, might shift just a tiny bit.
Due to what? Thermal changes.
Minor thermal changes, fluidic pressure, things like that. Now, standard decoding methods, like the nearest neighbor algorithm and Starfish, are extremely sensitive to this drift.
So if a spot moves too much, the algorithm just loses it.
It fails to recognize it as belonging to the same molecule sequence, and that drastically reduces the number of transcripts they can correctly identify, what they call their recall.
So the check-all decoder doesn't rely on perfect alignment. It seems to sort of embrace the messiness of the data. How does it work conceptually?
It works by exhaustive consideration and then statistical prioritization. So the check-all decoder doesn't just look for the closest spot in the next image. It considers all possible combinations of spots that could form a complete barcode across all the imaging rounds.
All of them. That sounds computationally insane.
It is. It then employs this complex scoring system to select the best non-overlapping set of codes. And the system prioritizes combinations that do two things. Minimize spatial variance, meaning the spots are as close together as possible, and maximize spot intensities, meaning the signal is strong.
It's like a very sophisticated, computationally intense form of molecular archaeology.
That's a great way to put it.
And what was the quantifiable payoff? How much better was it than the traditional nearest neighbor method?
The performance increase was a game changer. I mean, in non-corrected mode, the check-all decoder increased the total number of correctly identified transcripts by anywhere from 54% to 77% over the nearest neighbor method.
54 to 77%. That's not a marginal improvement.
Not at all. That's pulling more than half, again, as much useful biological data from the exact same raw set of images.
That is an amazing return on investment for software engineering. But as we've said, complexity always costs something. What's the major trade-off here?
The trade-off is computational expense, and it is steep. The older decoders are often basically instantaneous and require minimal memory. You can run them on a standard desktop computer.
But not check-all.
The check-all decoder, because of that exhaustive search function, takes significantly longer and demands massive memory resources.
So we're not just talking about a longer runtime. We're talking about moving from a task you can do on your desktop to one that might need a dedicated server farm.
Or access to high-performance computing clusters, yeah.
Yeah.
And that immediately creates a barrier to entry, right? It could potentially limit this powerful technique to only the best-funded research institutions.
It's a real issue.
It is, which is why acknowledging and trying to address that cost is so important. They tried to mitigate it by using Python's multi-processing features to spread the load, but the fundamental requirement is still there. It forces researchers to make a clear choice.
Speed and low cost versus maximum data extraction.
Exactly. Do you prioritize fast, cheap, but lower resolution data? Or do you dedicate massive computational resources to squeeze every last drop of biological information out?
I also noticed they built an error correction layer into checkall. This seems absolutely essential, given how easy it must be to lose a spot in one of those high hybridization experiments.
It's essential. So if the original DNA barcodes were designed with a sufficient Hamming distance,
Meaning they're different enough from each other sequence-wise?
different enough, yes, then the checkall decoder can actually decode error-corrected barcodes.
So even if it's missing a spot.
Even if a transcript is missing a fluorescent spot in exactly one of the imaging rounds, the system can still uniquely identify it just from the partial code. And this ability significantly increases your recall, pulling even more information from the data set.
But I imagine those corrected codes come with a caveat.
They do. They are inherently less accurate and more prone to false positives than codes that are decoded with a complete set of spots.
So it gives researchers a dial they can turn.
It gives them a dial, yes. You can prioritize a sheer volume of data, which is high recall with error correction, or you can prioritize absolute certainty, which is high precision without it. The checkall system provides that necessary flexibility.
Okay, so we've established how researchers are figuring out what molecular messages are being sent and where they're being sent from. Now let's shift to that second great challenge, mapping the physical wiring diagram itself. Let's talk about connectomics.
The field of connectomics really stands entirely on the shoulders of one person, the Spanish histologist Santiago Ramón y Cajal.
Working in the late 1800s, he's rightly called the father of the field.
He was a true visual genius. He used this silver nitrate staining method, the reazione nera, which, kind of by chance, stains individual neurons randomly.
And that allowed him, for the first time, to see the microanatomy of a single neuron. His drawings are just, they're works of art.
They really are. And that technique allowed him to reveal those microstructures and lay the foundation for the neuron doctrine, this revolutionary idea that the brain isn't a continuous mesh, but it's made of individual, discrete cells.
But here was the physical limitation.
Yes, Cajal's light microscope, even the best one of his era, simply could not resolve the fine structures of the synapse itself. They're just incredibly small.
And that structural gap required the great technological leap of the 20th century, the electron microscope, or EM.
Exactly. The EM breakthrough in the mid-1950s finally provided the resolution needed. It pushed visibility down to 0.1 to 0.0001 microns, a 1,000-fold improvement over light microscopy.
And that let scientists finally see the first synapse.
It did. It confirmed Cajal's doctrine and made modern connectomics possible.
But just like we saw with the spatial data, solving the resolution problem only made the scale problem even worse.
It became this epic Herculean task. I mean, the only complete connectome that has been mapped to this day belongs to a tiny roundworm, C. elegans.
Which has, what, 300 neurons?
Just 302 neurons. And mapping that circuit required Sidney Brenner and his team over 50-person years of painstaking labor.
50-person years.
Just reconstructing the circuit slice by slice from serial electron micrographs.
So if a tiny worm takes 50-person years, mapping a mammalian brain seems like an impossible dream, even with today's technology.
Unless something fundamentally changes, yes, we're still just drowning in data. Imaging a small rodent cortex, like from a mouse, could theoretically take over 600 years with traditional single-beam EM technology.
600 years?
And while new multi-beam microscopes are accelerating the throughput massively, we're talking several terabytes per hour now, the computational challenge of turning those raw images into a usable circuit map is still the single biggest bottleneck.
What's fascinating here is that even with faster imaging, the computation is the real problem. And it requires two critical error-prone steps: alignment and then reconstruction, or segmentation.
Right. Alignment, as you hinted at, is like trying to perfectly stack thousands of sheets of tissue paper where each one might be slightly stretched or rotated or compressed during preparation.
And you have to computationally correct for all of that.
You have to, just to trace a single neuron reliably across thousands of slices, and then comes segmentation.
Which is labeling every single pixel.
Labeling every single pixel according to the biological structure it represents. Is this an axon, a dendrite, a synapse, a glial cell?
And this is where the automated systems currently fail us, especially for tracing long-range connections.
They do. They generate these mistakes called merge errors and split errors. A merge error is when the system accidentally connects two separate objects. A split error is when it breaks one continuous neural process into two.
And the error rate is still too high.
Automated segmentation currently has an error rate of about 1 mistake per cubic micrometer of tissue volume. And while progress is being made very rapidly, that rate is still fundamentally inadequate for reliable, long-range tracing of complex circuits. We simply cannot rely on human tracers to proofread the billions of voxels being produced.
So the challenge is clear. EM gives us the resolution, but the proofreading and data generation are just unsustainable. This forces researchers to find new, really ingenious ways to label and manage the data.
One of the leading strategies comes from groups like 11 Bio. It's called PRISM.
PRISM, which is photoconnectomic reconstruction by iterative staining with molecular annotations.
Exactly. And this entire approach is designed to put information into the tissue that helps mitigate that EM proofreading bottleneck.
And they tackle the resolution problem in a really interesting way. Not by upgrading the microscope, but by making the tissue itself bigger.
Yeah, the now famous expansion microscopy. It's a really elegant physical solution.
It seems so simple.
It is. They forego the cost and complexity of super high-resolution EM by making the sample easier to resolve using a standard light microscope. They polymerize the brain tissue slice with gels.
Specifically sodium acrylate.
The same material you find in superabsorbent products, yeah. When they flush the tissue, the polymer swells, it strips away lipids, and it causes the tissue to physically expand by almost 2 orders of magnitude.
So you create this massive see-through ghost of the brain.
A ghost of the brain, exactly. And that solves the visualization problem, but you still need to know which neuron is which in that expanded ghost.
Right, you still have to tell them apart.
And that's where the protein barcodes come in. 11 Bio synthesizes these short peptide tags, the protein barcodes, and delivers them into the brain using harmless AAV viral vectors.
And each neuron takes up a random set of them.
A random unique subset of these tags. So every single cell gets a unique multicolored ID card.
That's brilliant.
Then they flood the expanded tissue with fluorescent antibodies that stick to those peptide tags, making the neurons glow in these distinct randomized color combinations.
And how many combinations can they get?
With their multiplex system, they can theoretically distinguish between over 100,000 unique color combinations. This unique ID code for each cell makes the tracing process so much easier.
Because you're not just relying on subtle grayscale variations in an EM image.
You have a built-in colorful signature that acts as a proofreading marker during the whole tracing process.
Have you ever wondered why we can't just sequence the connections? I mean, given the complexity of PRISM and EM, it seems almost too slow.
That is the revolutionary idea that's driving a new approach called BOINC.
BOINC. Barcoding of individual neuronal connections.
Right. This approach basically recognizes that the sheer complexity of the circuit requires a solution that can be processed in parallel at an exponential rate. So it converts the entire connectivity problem, a physical imaging challenge, into a problem you can read out rapidly with high-throughput DNA sequencing.
The power of sequencing in this context is just, it's hard to overstate.
It is the ultimate scaling mechanism for information. I mean, we are now routinely sequencing billions of nucleotides per day. And think about the economic scaling.
Incredible.
The cost of DNA sequencing has historically fallen at a rate of 15-fold per year. Compare that to the 1.4-fold yearly improvement rate of Moore's law for computing power.
So sequencing is just getting cheaper, faster.
It's rapidly becoming the cheapest, fastest way to map highly complex biological data.
So how is this molecular system actually implemented at the exact point of connection at a single synapse?
Researchers developed a system called SynBar for this. It's designed specifically for single synapse resolution mapping. The system involves 2 modified proteins, pre-SynBar in the presynaptic neuron and post-SynBar in the postsynaptic neuron. And these proteins are engineered to interact directly with each other across the synaptic cleft.
So they're mimicking natural synaptic adhesion proteins.
They are. And those modified proteins are carrying the crucial information payload.
The barcodes.
Yes, they're designed to bring specific paired RNA barcodes, a pre-RNA and a post-RNA together, but only when a synapse is actually formed.
And that interaction creates a stable complex.
It does, a stable complex that you can then isolate from the tissue and read out using high-throughput sequencing.
So the resulting sequence read literally tells you which presynaptic neuron barcode is physically connected to which postsynaptic neuron barcode.
Mapping the circuit one synapse at a time and completely bypassing the need for image reconstruction and human proofreading.
That is a radical departure from traditional connectomics. You're trading the incredibly high spatial detail of EM, where you can actually see the vesicles and the membranes, for massive, scalable connectivity information.
It's a trade-off, but it's a necessary one if you want to achieve scale. EM is slow, it's expensive, and it's bottlenecked by human proofreading.
Whereas SynBar is fast and cheap.
It's fast, cheap, and it's bottlenecked only by the power of your sequencer. So if your goal is the large-scale, comprehensive wiring diagram, SynBar offers an exponential advantage over every imaging technique that's currently available. It's a pure data solution to a structural problem.
But let's say you're a researcher who still needs that ultra-fine, ultra-structural detail that only EM can provide. You need to see the actual organelles, the vesicles, the membranes.
But then you are still stuck with the needle in the haystack problem.
Right. How do you find a tiny micron-scale structure inside a massive millimeter-scale block of tissue?
And that's where this computational strategy called Niwaki comes in.
Niwaki, which is Japanese for tree pruning.
For tree pruning or taming, and it contrasts sharply with the idea of random forests, which is the computational brute force approach of trying to automatically segment every single thing in a massive volume.
So the Niwaki goals is about sophisticated targeting.
It's about aggressively reducing the high-resolution imaging volume to only the structure of interest, saving time and terabytes of storage.
So how do they manage that physical and technological pruning?
They rely on advanced multi-shot volume SEM methods, things like array tomography or AT, and automated tape collecting ultramicrotomy, ATM.
And the critical distinction here is that you're not destroying the tissue after you image it.
Exactly. These techniques collect the serial sections onto a solid support, like a piece of tape, creating a permanent physical library of the tissue.
And that's the key. A library means you can go back and re-image specific spots.
Precisely. This library allows for reimaging and hierarchical scanning, so they can scan the entire slice at a low resolution, say 500 nanometers, to get the context.
Find the neighborhood.
Find the specific neighborhood where the critical synapse is located, and then go back and hit only that micron-scale spot with ultra-high resolution 3 nanometer EM.
To make that zooming in process efficient, they must need landmarks, right?
Absolutely. You need reliable guides that are visible at both light and electron microscopy scales. So they use endogenous landmarks,
Like blood vessels?
like the unique pattern of blood vessels or the tissue's natural autofluorescence. And they also use exogenous marks like near-infrared branding or NI-arbing.
So you're literally branding the tissue.
You are. And these marks allow researchers to perform correlative light and electron microscopy, or CLEM, ensuring they can precisely locate the structure of interest before they start the expensive high-resolution EM. It turns the whole imaging process from an exhaustive search into a targeted mission.
We've covered so much incredible new technology for mapping the brain. Now let's talk about the human payoff. Why are we doing all this painstaking petabyte scale work?
At the end of the day, it always leads back to understanding, treating, and eventually, we hope, curing complex brain diseases.
Let's look at schizophrenia as an example. The idea that this is an illness of disconnection. That isn't just a modern finding, is it?
Not at all. It's an ancient concept. Eugen Bleuler, who actually coined the term schizophrenia, which means split mind, he recognized it over a century ago as an illness of functional disconnectivity.
It's amazing how a century later, his conceptual diagnosis is being literally mapped onto physical wiring diagrams. This isn't abstract theory anymore. It's a measurable circuit imbalance.
It is.
What does modern neuroimaging tell us about this disconnection?
Modern techniques like diffusion tensor imaging, DTI, and functional MRI, fMRI, they provide concrete evidence of widespread brain disconnection.
And does the severity of this structural alteration correlate with how severe the illness is?
There's very strong evidence suggesting that the structural and functional alterations are often greater in patients with treatment-resistant schizophrenia, or TRS.
That's the most severe phenotype.
The most severe phenotype, yeah. Characterized by persistent, debilitating symptoms that just don't respond to standard antipsychotics. It's theorized that TRS is likely mediated by multiple complex circuit mechanisms that go beyond the simple hyperdopaminergia we usually associate with the disease.
So mapping these disrupted networks in TRS patients is critical.
It's crucial for understanding why standard treatments are failing them.
Let's pause and simplify that part again. What's the actual circuit mechanism that breaks down when we talk about this disconnectivity? How does a structural issue translate into the symptoms we see?
It really all centers on the failure of synchronized activity in the brain, specifically in what are called gamma band oscillations, or GBOs.
BBOs.
These are high-frequency synchronization signals. You can think of them as the metronome that allows vast, disparate groups of neurons to integrate information fluently and communicate efficiently across the brain.
And in schizophrenia, that metronome is broken.
The dysfunctional GBOs are linked to impaired communication between the system's accelerator and its brake.
So the core issue is that the glutamatergic drive, the accelerator, isn't communicating effectively with the GABAergic interneurons, the precisely timed brake.
That is the core model. The excitatory pathways, the accelerator, or the on-connectomic are driven by glutamatergic pyramidal cells. Their dysfunction is often linked to hypofunction of the NMDA receptor.
And the brake.
The inhibitory pathways, the brake or the off-connectomic, are driven by GABAergic interneurons, specifically a type called parvalbumin interneurons. If that delicate balance between excitation and inhibition gets disrupted, the neural circuit can't generate healthy GBOs.
And that leads to the cognitive and perceptual symptoms. So if connectomics can map these highly specific regional disconnections, if it can identify, for instance, that a specific TRS patient has a severe imbalance in GABAergic activity in their prefrontal cortex, it directly supports a shift toward precision medicine.
Precisely. It supports the whole concept of targeted therapeutics. Instead of relying on ubiquitous antipsychotics that modulate dopamine everywhere, future treatments can focus specifically on the localized fault in the circuit.
So that could mean what? Augmenting NMDA receptors or modulating GABA interneurons?
Exactly. Augmenting NMDA receptor function or modulating GABA interneurons to improve GBOs and circuit integration, but only in those specifically affected regions. This methodical mapping of excitatory and inhibitory circuit phenotypes in TRS is really seen as the most logical strategy for advancing customized drugs that actually address the root cause of the disconnection.
Moving from the clinic to the lab, all this advanced neuroscientific capability, especially when you're dealing with human tissue and data, it raises some profound ethical questions, doesn't it, about what we're creating and who we're studying?
Oh, absolutely. The development of human brain organoids, these mini brains grown in a dish from stem cells, is a stunning technical achievement. But as these models increase in size, as they last longer, and as they display more organized neural activity, we enter this really challenging ethical territory that requires careful, constant oversight.
What are the primary ethical concerns surrounding these increasingly complex models?
Well, the concerns usually revolve around a few key things: consciousness, sentience, and the moral status of the organoids themselves.
And the current oversight structures aren't really set up for that.
They're not. Committees like the EHSCROs focus primarily on the ethical provenance of the stem cell lines. Where did they come from? But as organoid complexity grows, that review may need to expand, potentially triggering discussions about what constitutes an embryo-like structure.
What do you think? I mean, how should we scientifically and ethically define consciousness in a petri dish? It feels like we're dealing with a concept that's just inherently vague. There's no clear metric.
That ambiguity is central to the ethical debate. If consciousness just means basic neuronal activity when you stimulate it, well, that's just basic brain mechanics, and it's ethically pretty straightforward.
But if it means subjective self-awareness?
If it means subjective self-awareness, the ethical stakes are raised dramatically. But crucially, most neuroscientists would suggest that complex consciousness requires the global integration and activation of cortical neurons across long distances.
So you need multiple, diverse brain regions working together.
And you need sensory input. Current organoids simply don't have that global structure or any sensory input.
So they lack the full complement of cell types and the necessary long-range connectivity.
Right. They lack what's needed for anything resembling complex subjective experience. So while the concern is valid and we absolutely have to keep monitoring it, the reality of current brain organoids makes that level of complex sentience scientifically unlikely right now.
So what's the path forward?
Transparency. Encouraging bioethicists and researchers to work together closely to anticipate and manage these issues as they emerge.
Let's pivot to the ethics of clinical data usage. We're collectively generating petabytes of neuroimaging data from these massive projects. But the question is, who is this data truly representing?
And this is a critical equity issue. The problem is that many of the foundational neuroimaging studies, especially the ones used to identify and validate critical biomarkers, have been focused predominantly on Western-educated and industrialized populations.
Which can reflect unintentional bias.
Biases related to race, status, geography, yeah. And that can lead to significant healthcare inequities globally.
Can you give us a concrete example of how optimizing a tool based on one population leads to diagnostic failure in another?
The most critical example involves Alzheimer's disease biomarkers. Amyloid plaques are a definitive hallmark, often detected with PE imaging.
Okay.
Well, the established diagnostic tools, the thresholds, the clinical standards for these biomarkers, they were predominantly optimized using Caucasian populations in North America and Europe.
And when those standards were applied elsewhere?
Studies quickly show that the amyloid accumulation patterns and the diagnostic thresholds identified in those Western populations did not consistently correlate with disease progression or cognitive decline in African or Asian populations.
Wow. So what does that mean in practice?
It means a diagnostic tool that was optimized for one group might lead to systematic misclassification, misdiagnosis, or delayed intervention in non-Western ethnic groups. It just exacerbates global health disparities.
So what are the strategies being pursued to ensure greater equity in this petabyte-driven field?
The effort has to be twofold. First, there's a huge push for significantly diverse representation in study populations to make sure the findings are broadly applicable across global demographics. Second, there's a demand for researchers to be highly transparent and openly disclose the limitations and potential biases in their study designs. You know, who is included and, just as importantly, who is not.
So the goal is to build tools that account for variability from the start.
Exactly. To develop neuroimaging protocols and even machine learning algorithms that are designed specifically to account for variability across different populations from the very beginning, rather than just reinforcing existing narrow biases.
That feels like a good place to wrap up the details.
I agree. Today, we've taken a really deep dive into the two colossal challenges defining modern neuroscience: molecular location and physical connection. We saw how spatial omics is decoding the molecular map.
Moving sequencing to the single molecule scale with systems like SMSBS.
Right, and conquering that computational standardization problem with pipelines like the check-all decoder.
And then we explored connectomics, mapping the neural circuits, moving beyond the heroic 50-year efforts of classic EM to these new barcoding strategies like PRISM.
And the revolutionary sequencing approach of SynBar, which leverages the exponential growth of DNA technology to map connectivity at scale.
It's clear that the future of brain science is fundamentally dependent on our ability to manage this data, to conquer that 1,400 terabyte problem with ingenuity, not just with brute force.
Right. And to help you recall the main components of this complex dive, let's use the mnemonic CAT for the three essential scales of brain mapping we covered.
Okay, CAT. So the C stands for connections.
This is the connectome, the physical wiring diagram. We saw how we can solve this with ultra-high-resolution imaging, like EM, made targeted by the Niwaki approach.
Or solved purely by molecular means using PRISM or SynBar.
Exactly. Then the A stands for alignment.
This is the computational challenge. It's the difficulty of solving that petabyte puzzle.
Whether that means stitching together thousands of physical slices, overcoming molecular spot drift with clever software like check-all, or developing new segmentation algorithms.
And finally, the T stands for transcriptome.
This is the molecular map. It's using those ingenious spatial barcoding methods like this SMSBS system to locate where every single molecular message is being expressed in 3D space.
Which is essential for understanding cell function in its proper context.
As Santiago Ramon y Cajal once observed, the brain is the greatest mystery. And this research, connecting bits of DNA barcode information and bytes of terabyte scale image data, it's key to solving the mystery that lives inside our own skulls.
And it has some pretty profound future implications.
It does. I mean, if tools like SynBar can map every connection and a system like SMSBS can decode every molecular message in its precise location, we are rapidly gaining the informational infrastructure we would need to even begin conceptualizing a complex goal like whole brain emulation.
Which makes the ethics of biological intelligence and issues like AI safety a critical and immediate consideration, not some far-off science fiction problem.
Exactly.
Thank you for joining again.